the recording has started, and yeah, just need to click on this. I know it's in Chinese. Yeah, and okay, now we can start talking.
You tell me when to start talking. When should I start talking?
At 12.
Now?
No, I'm going to introduce you.
Okay.
Yeah, okay.
Come in. I made a mistake. I went to escape.
Yeah, you exit from the screen sharing.
Okay. It's just I was trying to change this A up there. It's agenda, and I wanted to change the A. You know, get rid of the A. It's misspelled.
Okay.
But leave it there. It's okay.
Yeah. You want to see all the faces or only yours? Or only one?
Mine.
Okay. In this mode, you can see anybody who is speaking. Okay.
Okay. Thank you.
Thank you.
So it's you tell me when to start.
Sure.
Sure.
Okay, it's 12 past. Let's get started. Can you hear me, Dr. Hartman?
Yeah, I can hear you. Can you hear
Okay,
me?
yes, yes. Okay, great. Hello, everybody. Today, Dr. Hyman Hartman will give his last talk among the 3 talks about the origin of life. Dr. Hartman graduated with a Bachelor of Science degree in biochemistry from McGill University in 1957 and got his PhD degree in biochemistry at Columbia University in 1963. After that, he has worked at MIT, UC Berkeley, the University of Tel Aviv, and Harvard Medical School. He is now a research scientist in the Department of Biology at MIT and the director of the Institute for Advanced Studies in Biology at Berkeley, California. Dr. Hartman's research focused on the origin of life. He has published many books on this topic, including Search for the Universal Ancestor in 1987, Clay Minerals and the Origin of Life in 1986, and The Origin of Evolution of the Cell in 1992. Today, he will talk about the origin of life and von Neumann. Let's welcome our speaker.
Thank you, Yang Sheng. Today, I'm going to talk about von Neumann, a computational model for the origin and evolution of life. I'm going to start with Turing and the universal Turing machine, E. coli, the cell that founded molecular biology, von Neumann's self-reproduction, 4, cellular automata and self-reproduction, 6, Ising model and spin glasses, 7, computational origin of life, 8, evolution and machine learning, 9, the language of neural nets, and 10, back to Darwin. So let's start. So here are the 2 men who in 1946 approximately brought gifts to the world of life and a lot of other areas from, guess what? Mathematical logic. It was from mathematical logic that Turing brought the universal Turing machine, and von Neumann brought the self-reproducing automaton. So let's see what that happens. On Turing's 100th birthday, Sidney Brenner, who is, I consider, the greatest molecular biologist of the 21st century for sure. And he wrote a paper in Nature called Life's Code Script. Turing machines and cells have much in common, argues Sidney Brenner. Turing ideas were carried further in the 1940s by mathematician and engineer John von Neumann, who conceived of a constructed machine capable of assembling another according to a description. A universal constructor with its own description would build a machine like itself. To complete the task, the universal constructor needs to copy its description and insert the copy into the offspring machine. Von Neumann noted that if the copying machine made errors, these mutations would provide inevitable changes in the progeny. So let's begin with Turing. Turing, as you well know, is the inventor of the Turing machine. This comes directly out of mathematical logic, and that is of great interest. So a Turing machine is a mathematical model of computation that defines an abstract machine that manipulates symbols on a strip of tape according to a table of rules. Despite the model's simplicity, given any computer algorithm, a Turing machine capable of implementing that algorithm's logic can be constructed. The machine operates on an infinite memory tape divided into discrete cells. The machine positions its head over a cell and reads or scans the symbol there. Then, based on the symbol and the machine's own present state, in a finite table of user-specified instructions, the machine first writes a symbol, digited or a letter from a finite algebra alphabet in the cell, then either moves the tape one cell left or right. Then, based on the observed symbol and the machine's own state in the table, either proceeds to another instruction or halts computation. The universal Turing machine. It is possible to invent a single machine which can be used to compute any computable sequence. If this machine, U, is supplied with the tape on the beginning of which is written in a string of quintuplets, separated by semicolons of some computing machine M, then U will compute the same sequence as M. So here's a universal Turing machine. You have a code number of a Turing machine. You have the input to M and the output, which tells you what to do when you're in a state and you're observing a symbol. And here is sort of a diagram of the infinite tape. Now keep that in mind. The world of a Turing machine is an infinite tape of squares. And it has a state transition diagram, which is related to a finite state automaton. The Turing machine uses the description on the tape, passes it into a universal Turing machine, and then the calculation is made on this tape. This is sort of, again, just a very simple view of a finite state machine viewing a one. It's either going to go right or left and then change its state. So it's very important to note that there's a finite state automaton and that reads and does it all. Now, related to a set of computers, of which the Turing is the highest level, there is a Chomsky hierarchy of language related to computational machines. So a finite state automaton has a regular language. Push-down automaton has a context-free language. The memory system in the push-down automaton is not infinite. A linear bounded automaton has a context-sensitive language, and then Turing machines has recursively enumerable language, and it has an infinite tape. So all these machines are related to how much memory they have. Now I want to stop and talk about the cell that founded molecular biology, E. coli, and give you a very quick summary. This is E. coli. It's approximately 1 micron in length. This is when you explode E. coli, this is the DNA in E. coli, one single molecule. All that molecule is one single strand of circular DNA. Now, here's what happens. The DNA encodes the description of E. coli, and it is embedded in these RNA, which can be read from the DNA and becomes involved with the ribosome in translation. So you start off with DNA, a segment of DNA is transcribed into messenger RNA. Messenger RNA goes into the ribosome and you make your peptides. And reading the messenger RNA 3 nucleotides at a time. Here is the ribosome, the actor. Here is the messenger RNA going in, and it's being read by tRNA every 3 nucleotides. that puts an amino acid onto a growing chain of peptide. And so you translate the messenger RNA into the amino acids and into the peptides, growing one amino acid at a time. The ribosome occurs in a large subunit and a small subunit. Here is another actor of T4, of E. coli. This is the virus, T4. It's a very famous virus. It was worked on here at Caltech by Max Delbruck, and a lot of E. coli, a lot of molecular biology was worked out on T4 viruses. Here is a T4 virus injecting its DNA into an E. coli, and it's just going to make more T4 until the cell bursts open and puts out the T4 viruses into the media. Now, the thing you should recognize here is the DNA that's injected into the E. coli is coding for T4, all its proteins, and also for its DNA. So what is E. coli? It's 4 million base pairs in its DNA, 4,000 proteins, 4,000 messenger RNAs, 2 ribosomal RNAs, 20 plus tRNAs, and DNA is 1.36 millimeters. This is how you grow it. This is a minimal media. It has M9 salts, which is sodium phosphate, potassium phosphate, and ammonium chloride. And it also has glucose and magnesium sulfate, calcium chloride, and water. And that is the medium from which E. coli is now going to grow. 4,000 peptides, et cetera, et cetera. Now we come to von Neumann. Von Neumann is the center focus of this talk. And in a letter he wrote to Norbert Wiener, they were discussing, this is in 1946, there's a 6 missing. In 1946, they got together, and at that time they were saying, wow, we have this new computational model for everything. And the question is, should we attack the brain or should we attack something else? Norbert Wiener took the brain. He was going to attack the brain. And he built a big team at MIT with McCullough Pitts, etc. And von Neumann said, this is too complicated. We don't know what's going on in the brain. I'm going to pick a typical bacteriophage, which can be multiplied at will and hence counted by the colonies it forms on a suitable substrate as reliably as elementary particles can be counted by a Geiger counter. Is a phage that is parasitic, I think on Bacillus coli. It has been extensively worked with, e.g. by Delbruck at Vanderbilt. And Delbruck was at Vanderbilt before he came to Caltech. And here's what he says. Study the main types of evidence, physiology of viruses and bacteriophages, and all that is known about the gene-enzyme relationship. Genes are probably much like viruses and phages, except that all the evidence concerning them is indirect, and that we can neither isolate them nor multiply them at will. I am going to then think a great deal about self-reproductive mechanisms. I can formulate the problem rigorously in which Turing did for his mechanisms, okay, for Turing machines. I can show that they exist in this system of concepts. I think that I understand some of the main principles that are involved. I want to fill in the details and to write up considerations in the course of the next 2 months. I hope to learn various things in the course of this literary exercise, in particular, the number of components required for self-reproduction. My rather uninformed guess is in the high 10,000s or in the 100,000s, but this is mostly unsafe. Besides, I'm thinking in terms of components based on several rather arbitrary choices. At any rate, it'll be necessary to produce a complete write-up before much discussing it is possible. In 1948, von Neumann did his write-up in a Hickson symposium here at Caltech, and everyone should read that write-up. Here's what he said in the write-up. There are a number of components to this self-reproducing machine. D is the tape that has all the instructions of how to make the machine. B is a duplicator that will copy that tape and add it to the new automaton. B is also related, and this is sort of ad hoc, is the RNA polymerase, which essentially gives an instruction to the constructor, which is the ribosome, to make protein. And this protein now is going into making the new automaton. So the D's are the instructions, B is the duplicator, and then at the end of having made a new automaton, attaches the full instructions onto this copy, onto the new automaton. So it's A, the constructor, B, the duplicator, C, the controller, which is something to do with, and this now goes to the instructions. This was Freeman Dyson's interpretation of what all this was. So the component A is an atomic factory, which collects, it said it's a ribosome. B is a duplicator, an automaton, which writes an instruction and copies it. C is a controller, which is hooked up to A and B. C is given an instruction. It first passes the instruction to B for duplication, and then passes it to A for action. And D is the written instruction containing the complete specifications which cost A to manufacture the combined A plus B plus C. And here is what von Neumann said. Von Neumann's analysis showed that a structure of this kind was logically necessary and sufficient for a self-reproducing automaton, and he conjectured it must exist in living cells. 5 years later, Crick and Watson discovered the structure of DNA, and now every child learns in high school the biological identification of von Neumann's 4 components. D is the genetic material, DNA. A is the ribosomes, etc. B is the enzymes, RNA and DNA polymerase, and C is the repressors and derepressors. Okay, so far as we know, the basic design of every microorganism larger than a virus is precisely as von Neumann said it should be. This is Freeman Dyson in his book on the origin of life. So here is his model, his first model. To model self-reproduction at even the most abstract logical level, we need to place a kinematic automaton in an environment which can interact. This environment will be composed of the same stuff as the automaton is made of, namely various primitive elements just listed. Imagine an infinite body of liquid with infinite many copies of each kind of part distributed in random over its surface. There are switches, delays, kinematic elements, cutting elements, fusing elements, rigid elements, and sensing elements floating on the surface. Imagine that they are moving back and forth, random motion after the manner of molecules of a gas. On this arrangement, an indefinite supply of parts is available to any kinematic automaton which floats on the surface of the lake. We call this environment, together with any kinematic automaton, it contains a kinematic system. Now I just want to introduce you to cellular automata. And the question is, here is cellular automaton. And this is the simplest one. It's basically a two-dimensional array of it's a crystalline array of squares that goes on infinitely in all directions. Here is what a neighborhood, a local neighborhood looks like. And in each square, there is a 1 or a 0. And there is the rules that says, if there's a certain number of ones around this one, it will remain a 1, or if there's more than a number of ones around this one, it will die, etc., etc. This is the cellular automaton. And every time step, that rule is done for every single square. Who invented cellular automaton? Johnny von Neumann. They were invented in the 1940s by John von Neumann and Stanislaus Ulam at the Los Alamos National Laboratory. Through apparently simple, some CAs are universal computers, that is, they can do any computer-capable computation. Now here is what von Neumann now changed from that kinematic thing to the cellular automaton. What he had in every square was a finite state automaton, which has a certain number of states. It could have 2 states, 0 and 1, or it could have n states. And what happens is the tape was, as you can see, similar to the tape of Turing. It is a one-dimensional, it's not necessarily infinite. It's a finite tape which can be extended indefinitely. Each square now bears the symbol 0 or 1. And therefore, this instruction now turns on a finite state automaton in a certain state. And now this is his construction. His construction is you have D, which is the tape, and the logic box, which interprets that tape, and then there's a construction arm that turns on a finite state automaton in each square. In each square, there's a finite state automaton. Now, von Neumann's built this machine, in which he embedded the tape and the finite state automaton that was reading the tape, completely did it in a 2-dimensional cellular automaton. So all of those things were already embedded in the 2-dimensional cellular automaton. In each square, there was a finite state automaton which was at 0. It was not active, and it was being activated by reading the tape, etc., etc. Now, the number of states von Neumann needed in order to build his self-reproducing machine, he had to make all these things that he talked about, like from NAND gates or something like that. Anyways, what happened? Here, he had to build all these things that he talked about. And he used 29 state automaton, and he needed 130,622 cells. Todd said, I can do it with less. I can do it with 8 states in each cellular automaton with symmetries. And yes, it's computation and construction universal. He needed 283,126,588 cells. And then we go down to Banks that used only 4 cells, and he needed 100 billion cells to do it. So basically, trying to build a cellular automaton that was self-reproducing was not the way to go. Now, here's a cellular automaton as we now see them. And this was a cellular automaton that you could find in Wolfram's book on a new kind of science. And here's the various rules. These are nearest neighbor rules. So if there's, the colors are black is 1 and white is 0. And so if there's 2 zeros and you're a 0, you stay a 0. If there is a 0 on your left side and a 1, I'm sorry, and 1 on your right side, you will become a 1. And then you can go through these laws, etc. And here's the pattern. Every time step is the next layer. So this is a one-dimensional cellular automaton in which every time step is now recorded. And now you see it giving you a rather interesting pattern. Now, in the two-dimensional cellular automata, there are 2 kinds of neighborhoods that are used. One is the von Neumann neighborhood, the other one is the Moore neighborhood. And the Moore neighborhood has 8 neighbors, and the von Neumann neighborhood has only 4. Now, I would like to introduce you to the next interesting character in cellular automata, and this was John Conway, brilliant mathematician. He was a delight to be with, and he would always have something interesting to say. And he invented the game of life. Now, the game of life finally put cellular automata on the map. The universe of the game of life is an infinite 2-dimensional orthogonal grid of square cells, all of which is in one of 2 possible states, live or dead, 0 or 1, populated and unpopulated respectively. Every cell interacts with its H neighbors, which are the cells that are horizontally, vertically, or diagonally adjacent. At each step in time, the following transitions occur. Any live cell with fewer, that means it's a 1, with fewer than 2 ones in its neighborhood dies. Any live cell with 2 or 3 live neighborhoods, 2 or 3 ones in its neighborhood, lives on to the next generation. Any live cell with more than 3 dies, as if by overpopulation. Any dead cell, which means 0, with exactly 3 live neighbors becomes a live cell, as if by reproduction. And so these are little things that you could make out of things. You start off with 4 ones and all zeros in its neighborhood. It remains absolutely stable. Here is the one that blinks. Every time step it goes up, goes down, goes up, goes down. The one in the right-hand corner is the most famous of all. It's the glider. So here you'll see that the glider is 3 squares away from the edge. And now it moves, and now it's 2. So this glider moves. And it was out of gliders and glider guns that they made the universal Turing machines, and also I think more recently, the constructing machines. Now, the interesting point about the game of life, that with very simple rules and interesting initial conditions, you can get enormously complicated behavior. And this was taken up by Fredkin, who designed a philosophy called digital philosophy. And digital philosophy is one type of digital physics. It's pan-computationalism, a school of philosophy which claims that all physical processes of nature are forms of computation or information processing at the most fundamental level of reality. And I joined the Fredkin group in 1985 or 86. And I worked with Tom Toffoli and Norm Margolis, and Pablo Tamayo was a brilliant graduate student, and Gerard Vishniak, a great physicist. And this is sort of an interesting story, because when I was a graduate student, when I became a graduate student from McGill, I decided I was going to be a crystallographer. And so I came to McGill, to Columbia, and I started to become a crystallographer, because the big problem at that time was, what is the structure of a protein? And the only way to work on that was through crystallography. So I took a course at Brooklyn Polytechnic with Dr. Van Kuchen. And one of the lectures he gave there was, holy moly, there was space-time crystal groups. That means space groups of crystals. There's 230 space groups of minerals. And then there was this new guy who had published a paper on space-time groups, 4-dimensional space-time groups. This is not relativity. This is not Minkowski space. This is just Euclidean space. So this goes back, and I said, holy moly, maybe that's the answer to life and the origin of life. Because in 3 dimensions, minerals, the inorganic forms of interest, are all space groups. Why not, if you have a space-time crystal, that is a living system? And this is an article by my friend Tom Toffoli, and he's talking about the space-time crystal. That was sort of interesting. Now, here is a finite state automaton in each one of these squares. And this is a model. It's the Greenberg-Hastings model. And it's based on a diffusion term, which controls the transition of quiescent, et cetera, states. You start off with a dead state. A 1 comes in, and immediately it becomes a 1. It comes alive. Then it goes in the next time step to a refractory state, and then in the next time step, it goes back to the 0 state. It's a small oscillator, basically. Now, if you set these things up in the as initial conditions, the top part are all in the 1 state, and the 2s down below are, I believe, in the refractory state. They essentially evolve out into a spiral. So the 0 is the quiescent state, black is the active state, and the stripes are the refractory state. So these turn out to have these little singularities. These are, having an active state right next to a refractory state over 0 states, and then 1 and whatever, 1, 0, 2, 2, 0, 1 on top of each other. And then you put them down randomly. Okay, you put down 0s and 1s randomly. And very quickly, they will start forming these beautiful spirals. And this is a great model for something that was done a la Turing. And this is the Belousov-Zhabotinsky reaction based on reaction and diffusion systems. And this was worked out by Turing. It's a very famous system of reaction and diffusion systems that is used all the time in embryology. And here is the same systems of circles, etc., on a surface of a platinum crystal. And this was done by a Nobel Prize winner on carbon monoxide and oxygen, forming these beautiful patterns. So what this meant is that cellular automata now can really model really complex chemical systems. And drawing spirals, Gertl Ertl and his colleagues discovered these patterns of carbon monoxide and oxygen, light and dark, on platinum surfaces and also figured out how to control them. Ertl's 2007 Chemistry Nobel Prize recognizes his contributions to the field of surface chemistry. So that means that a cellular automata can really deal with surface chemistry. So here's my epiphany. Why do your chemistry on a two-dimension? Why not do your chemistry on a two-dimensional cellular automata and then just replicate it in time, but replicate it also in space in the third dimension? And therefore you've solved the problem of a replicator. And do the rules have to be deterministic? I turn to a probabilistic cellular automata, the famous Ising model. So this is statistical mechanics and the Ising model. Now, the Ising model deals with spins. So if all the spins are interacting ferromagnetically, they are all in the same direction. If they're acting antiferromagnetically, they go in opposite directions. And so here you see these spins either up, or spins are down, or they're alternating if they are antiferromagnetic. So there is forces on the bonds between the spins, which are ferromagnetic and antiferromagnetic. And if you put them down at, and this is the normal thing, if they're just ferromagnetic, this is the interaction, Jij, between two neighboring spins. And there's also a conjugate field. Here's what happens when you go through a critical point. The Ising model is one of the most important models in phase transitions. And the Ising model here is going through the phase transition. When it goes down to zero temperature, it is going to be all up, okay, or all down, depending upon its conjugate field. If there is a slight perturbation in the up direction or a slight perturbation in the down direction. Now, the Ising model also works on binary alloys. So one of these alloys, one of the ions may be iron and the other one may be aluminum, or one of the may be iron and the other one is manganese. Here, these are not binary alloys. These have more than two ions, but it's the same thing. These are solid solutions. And these are also catalytic surfaces. So this is what happens in the case of just having a ferromagnetic interactions with a critical field being either up or down, the conjugate field up or down. And then you see that there are two minima in free energies. So that means you have an order parameter and there is a free energy, and the order parameter is at a minimum at these places, and therefore the free energy is also at a minimum. So at this particular junctures, this is a phase transition with one bit of information. Now, a spin glass, with the Ising model, the so-called spin glasses can be described by the usual Hamiltonian. It's the same one I gave you. Forget about, there's no conjugate field here. Where the S variables describe Ising spins, but they could describe two ions in a binary alloy. While the JIK are taken from random distribution. So they're not all ferromagnetic and they're not all antiferromagnetic. For spin glasses, a typical distribution chooses antiferromagnetic bonds with probability P and ferromagnetic bonds with 1 minus P. These bonds stay fixed or quench even in the presence of thermal fluctuations, When P is equal to 0, there's no antiferromagnetic bonds. The original Ising model exists. But here is what happens. When we look at the free energies and the order parameters, you can see that in the spin glasses, you have a lot of minima. And this now means that this can now calculate a lot more than the ferromagnetic Ising model. And the ground states are very complex. And in order to calculate these things, you can calculate the computational complexity of an Ising spin glass with Ising spins, and the problems of computing the magnetic partition function and finding a ground state are studied. In a finite two-dimensional lattice, these problems can be solved by algorithms that require a number of steps bounded by the polynomial function of the size of the lattice. In contrast, there are non-NP cases, like if you have a magnetic field or in 3 dimensions. And there's this interesting thing that the Ising model and combinatoric optimization and complexity theory it has been used to solve the Chinese postman problem, which is a variant of the traveling salesman's problem. So many years ago, in fact, in 1983, Hartman, following Karen Smith, who was the man who proposed that clays were the original organisms, has proposed a logically consistent scheme of the primitive replicating state is not chains of RNA having linear arrays of nucleotides, bases, but layers of silicate minerals having 2-dimensional array of different ions. From the university class of view, this proposal is totally equivalent. The crucial stage requires conjugation, symmetry, and random interaction with the environment. I have a personal preference based on Occam's razor for the present model, but of course cannot exclude any other scenarios. And this was, of course, his model of Anderson's for the origin of life. Now, something really has happened in recent years. You remember that you have Turing machines with instructions, and then you can go to the universal Turing machines. Well, now we have a Hamiltonian mimicking machine, and therefore you could have various kinds of Hamiltonians that map into a universal Ising machine. And so the Ising machine, given that you can have these various kinds of interactions from ferromagnetic, antiferromagnetic mixtures, whatever, does map into a universal one. So that's interesting about the Ising model. So therefore, I have modeled, of course, this model of has been modeled on clays and the origin of life, the experiments. This is a paper I published 2 days before I came out here. So therefore, I was not going to discuss clays and the origin of life here. But I suggest, given what I've told you about what I consider the computational origin of life, which I've just ended, that you should consult this paper. It's on the web. And if you have any questions, please let me know. Now I want to talk about the evolution and machine learning. Now, we are now living in a very interesting time where an artificial intelligence has generated 2 interesting subcases. One is machine learning, algorithms with the ability to learn without being explicitly programmed. That's probably approximately correct of Valiant. And deep learning, subset of machine learning in which artificial neural networks adapt and learn from vast amounts of data. This has recently been used to solve one of the deepest problems in molecular biology, which is, given the sequence of amino acids, can you determine its three-dimensional structure? And for years and years, one was trying to do that. And now with deep learning, one has solved, or has almost solved the problem. Now, what is deep learning? Well, deep learning starts off with an input layer of data, and then it goes through this hidden layer, and then there's an output layer. And in this inner layer, it abstracts various sub things and then puts them together and gives you the output. Here's the case, sorry about that, of the recognition of George Washington by a deep learning neural network. And essentially, it first identifies edges in the picture, then it identifies combination of edges in the picture, then it identifies features in the picture, and then it identifies combination of features, and then you get the output of George. So it's neural networks that are now of interest. And now there's an interesting relationship that's growing between these evolution of these deep learning. For instance, in AlphaGo, they had one machine interacting with another machine, and they were learning how to play the game of Go. And it evolved into a final game that and rules that was unbeatable. It beat the world's thing without any input. They were just playing against each other. And so there was a evolution going on in this AlphaGo situation. So there is now a large group of people in artificial life that are trying to study artificial evolution in deep learning networks, playing them off against each other, etc. And so they're running into the problem for a potential of open-ended evolution. Natural evolution gives the impression of dealing to an open-ended process of increasing diversity and complexity. If our goal is to produce such open-endedness artificially, this suggests an approach driven by an evolutionary metaphor. Ultimately, we hope to inspire researchers to be able to interchangeably use evolutionary and gradient descent-based machine learning methods to approach the design and creation of open-ended systems. Understanding the open-ended complexity of the natural world is one of the greatest outstanding problems in evolutionary biology, and emulating such complexity is one of the answers. This, of course, is the answer I gave in my previous paper, my previous talk, that ontogeny recapitulates phylogeny by starting simple and just building one step at a time, and it accumulates into the phylogeny of a very complicated entity. Now, the language of neural nets, because now we're talking about neural nets as the model of what is being computed in deep learning. Now, von Neumann wrote a very famous book called Computer in the Brain. This was the last book he wrote, and this is the last paragraph in the book. And here's what he says. We have now accumulated sufficient evidence to see that whatever the language the nervous system is using is characterized by less logical and arithmetical depth than we are normally used to. The following is an obvious example of this. The retina of the human eye performs a considerable reorganization of the visual image as perceived by the eye. Now, this reorganization is affected on the retina, or to be more precise, at the point of entry of the optic nerve by means of 3 successive synapses only, in terms of 3 consecutive logical steps. The statistical character of the message system used in the arithmetics of the central nervous system and its low precision is also indicate that the degeneration of precision described earlier cannot proceed very far in the message systems involved. Thus, logics and mathematics in the central nervous system, when viewed as languages, must structurally be essentially different from those languages which our common experience refers. And this is the problem we have with deep learning, because in the game of Go that was won and it was brilliant, we could not communicate with the machine as to what the hell it was doing and why it was doing. So the next big thing in deep learning is to try and figure out what language they're using and what code. So this again brings us back to the traditional Chomsky hierarchy. Here it is now for the case of deterministic machines. We now have to work out a similar hierarchy for probabilistic neural networks. Now, just to make there is someone else who has been studying biological evolution as a form of learning. And this is Leslie Valiant, who has written a book on the subject. And his basic idea is that learning is not theory driven. It's driven by induction. And the question is, how do you induce regularities in your environment without having a theory? And he has a very interesting, he has a very interesting model, and he is trying to essentially point out that it adds to the Darwinian model. We formulate a notion of evolvability that distinguishes function classes that are evolvable with polynomial bounded resources from those that are not. The dilemma is that if the function class, say the expression levels of proteins in terms of each other, is too restrictive, then it will not support biology. While if it's too expressive, then no evolution algorithm will navigate it. Okay, so now having originated life and now talking about how life may evolve, the next and final thing I want to talk to you about is why this is of interest to people studying evolution. Back to Darwin. In the last lecture, I discussed Darwin's one diagram in his book, Origin of Life. And I never discussed natural and sexual selection. So this is an interesting point, because this was a great debate between Darwin and Wallace. And Wallace pointed out that the term natural selection was causing misunderstanding about naturalists. He thought that the reason for this arises almost entirely from your choice of the term natural selection. And so constantly comparing it in its effects to man selection. And also you're frequently persifying nature as selecting, as preferring, as seeking only the goods of the species, etc., etc. To the few, this is as clear as daylight and beautifully suggestive, but to many, it is evidently a stumbling block. I wish, therefore, to suggest to you the possibility of entirely avoiding this source of misconception in your great work, if not now too late. Also, ineffectively by adopting Spencer's term, which he generally uses in preference to natural selection, survival of the fittest. This term is the plain expression of the fact Natural selection is a metaphorical expression of it, and to a certain degree indirect and incorrect, since even personifying nature, she does not so much select special variations as exterminate the unfavorable ones. Now, this is me talking. It has always been a puzzle. What is the function of the brain? Take any other organ of the body, and the answer is immediate: stomach, digestion, kidney filtration, heart pumping blood, and so on. Natural selection can obviously operate on these organs. But the brain, what is its function? What the brain does, among other things, is to create a model of an animal's world. It is clear that natural selection can operate on an animal's model. If an animal mistakes a predator for an innocuous intrusion into its world, then both the model and the modeler become lunch. However, it is also clear that the model built by the brain of an insect selects a flower. The model built by the brain of a female bird of paradise selects the impressive beauty of her consort. The major problem that one faces is, why do models generated by organisms as different as birds, naturalists, give rise to illusions in a similar manner as in butterfly mimicry? In fact, the whole enterprise carried out by naturalists assumes sufficient overlap between the worldviews of the organism studied and that of the curious human observer. This, in a deep sense, was Darwin's greatest insight. Natural selection has a psychological component. This fact is not simply the result of evolution, it in turn directly affects evolution itself. This psychological factor belongs to a field called meta-evolution, or to be more precise, the evolution of evolution. In summary, Darwin believed in a combination of ecological and psychological factors in natural selection. Wallace believed in the ecological factor, survival of the fittest. The best way to reconcile this situation would be as follows. In the early ecosystems, there was very little psychology. As the ecosystem became more and more complex, an organ of selection, the brain, appeared, which now selected. And so gradually psychology displaced ecology as the dominant selective factor. With man, the brain so dominated ecological factors that the world is now threatened. Thus, natural selection evolved from ecology to psychology. And that is the end of the thing. And you can see if I have gone through the agenda correctly.
Thank you very much for the fabulous talk, Dr. Hartman. Yeah, we have time for one question.
No questions?
Does anyone have questions?
Okay, can I just make a little addendum to what I just said?
Sure.
In 1946, von Neumann wrote that famous letter that I mentioned earlier. And here's what he said in the beginning. In trying to understand the function of automata and general principles governing them, we selected for prompt action the most complicated object under the sun, the brain, literally. In spite of its formal complexity, this subject has yielded very interesting information under pressure of efforts of Pitts and McCulloch, both of whom I knew. Pitts, Wiener, and Rosenbluth. I did not know Wiener and Rosenbluth. Our thinking, or at any rate mine, on the entire subject of automata would be much more muddled than it is if these extreme bold efforts, with which I would like to put on one par the very unneurological thesis of A. Turing, that means he's putting that together with Turing, had not been made. Yet I think that these successes should not blind us to the difficulties of the subject, difficulties which I think stand out as more as ever. And then he picks, I want to pick an organism, I picked the bacteriophage, and he wants to study the gene enzyme relationship. And then he goes into self-reproduction and the Hickson Symposium. So what I basically said in my very first lecture with Aristotle, that first of all, we discovered and talked about the vegetative soul, the soul of reproduction and catalysis. And now, as we end up this talk, we are studying the brain and perception. And God knows if we'll ever get to the rational soul. This, of course, was Aristotle in the first lecture. And now I am done.
Thank you. Thank you for giving the addendum. Okay, does anyone have a question? Well, if no questions, let's thank our speaker again. Thank you, Dr. Hartman, for your 3 talks. Okay, we'll meet after the new term begins.
How do I stop it?
Well, I can stop it for you.